Thank you so much for a very nice introduction, and thank you so much for being here to listen to my talk. Today, I will present my lab's research on the topic of explainable AI, a subfield of AI focused on enhancing the interpretability of complex machine learning models, particularly in the field of biomedical sciences, of course. All the work was primarily done by an incredible group of passionate researchers pursuing PhDs in computer science at the UW, including the MSTP students concurrently working toward MD and PhD degrees in computer science. We work on a broad spectrum of problems in the ABC fields, including AI. We aim to develop explainable AI principles, theories, and techniques. B, biology. We try to identify cause and treatment of challenging diseases, our current focus being cancer and Alzheimer's disease. And C, clinical medicine. Our goal is to develop and audit clinical AI models. Today's talk will consist of 3 parts. So the first part will focus on the concept of explainable AI in the most traditional sense, and many examples illustrating how this traditional explainable AI can enhance biomedicine. The second part will focus on how explainable AI can pave the way for the creation of new research areas. So this will provide insights into how explainable AI need to evolve to effectively address a broad spectrum of real-world problems in biomedicine. Next, I will outline some of my future research directions. So in this talk, we won't delve deeply into any specific project. Instead, we will explore many use cases of various explainable AI techniques, and then show how these techniques can potentially revolutionize biomedical fields. Notably, these techniques are generalizable. So one of the ideal outcomes would be that you apply some of our methods to your own research problems in the field of computational biology. Okay, so explainable AI is probably the term I use the most frequently up to now. So what is it? So the key goal of explainable AI, or XAI, is to understand why a certain output was generated from a machine learning model, such as dementia onset risk prediction model. When we have a set of patient features and a reliably trained machine learning model, we can predict this kind of clinical outcomes accurately. However, in the traditional machine learning paradigms, we couldn't answer very natural questions of why a certain prediction was made. It is because with large modern data sets, the best accuracy is often achieved by complex models even experts struggle to interpret. The idea of explainable AI is to explain the output of a black box machine learning model based on the amount of contributions of individual input features for each individual prediction. And my lab has developed a series of explainable AI techniques. One notable example is the SHAP. It was led by my former student, Scott Lernberg. It's a general framework that's applicable to any model and has desirable theoretical properties that set it apart from other methods in this domain. And just to give you a very basic idea of how it works, the core question here is how much adding a particular feature I changes the model output. This, of course, depends on which other features the model also considers. So we could evaluate all possible subsets of features and average the change across them. And this approach, grounded in cooperative game theory, ensures that these SHAP values, feature importance scores, or Shapley values, satisfy the fairness axioms. So these are desirable properties as an explainable AI method or feature attribution method. So we guarantee that our solutions meet these important theoretical standards, and this point is really important. And the challenge, though, lies in the complexity of properly removing features from a machine learning model. Additionally, considering all possible subsets of features, of course, would introduce a significant computational complexity. So this led us to do both theoretical and empirical research in machine learning to make it more practical and feasible for real-world applications. So for example, we established a unifying framework where we showed that SHAP unifies a large portion of the XAI literature with 25 distinct methods following exactly the same basic principle, which is explaining by removing features. Turned out that all these methods, they work by removing features in slightly different ways. And then this kind of work helps us to understand what happens when these models fail or succeed, and allows us to improve and develop new XAI techniques that are accurate. To address the computational complexity, we developed new optimization frameworks for SHAP in largely two directions: model agnostic approaches, and then also model specific approaches for various types of popular machine learning models, including ensemble trees, neural networks, deep SHAP, and transformers. And our study also shows that SHAP is very effective in various tasks, such as adaptive feature selection, accurate estimation of global feature importance, and then also interpretation of unsupervised models. And as it turns out, developing the SHAP framework didn't solve the problem at all. It just opened numerous research avenues in the field of explainable AI. Okay, so before I put you to sleep, let's quickly shift gears to discuss our journey in biomedical fields. Okay, so using one of these developments that was a tree SHAP published in 2020, we revealed individualized risk factors, so per patient, per subject, risk factors of chronic kidney disease, all-cause mortality, and several other clinical conditions. So it's all detailed in our paper, Nature Machine Intelligence. So these results would provide new insights in terms of what are important for these clinical conditions. There were the things that we already knew, but also new things we haven't really known, and that we worked with domain experts, specifically my collaborators at UW Nephrology, to really understand what they mean. So in another paper, which is the first applied paper on SHAP, we, Scott and I, tackled the question of whether explainable prediction can improve the clinician's ability to predict clinical conditions in anesthesia care. Surgery and anesthesia are integral part of healthcare, but also pose considerable risk of complications and death. So we focused on unwanted physiological condition that's called hypoxemia, meaning low blood oxygen level that causes a serious patient harm. If hypoxemia can be predicted before it occurs, say even 5 minutes, then anesthesiologists can take actions to proactively prevent hypoxemia and minimize the patient harm. So for that, we developed the prescience method that predicts the near-term risk of a hypoxemia during surgery in real time and explains the factors that led to that risk. So here is how it works. Prescience outputs the risk prediction and its explanation, which shows a set of features that increase the risk, purple-colored ones, and then decrease the risk, green-colored. And the plot at the bottom is equivalent to rotating this feature explanation bar at certain time point by 90 degrees, and then stacking these explanations for each time point horizontally. So you can see that the risk trend, that the large increase in risk at the current time point was driven by titer volume, meaning a drop in the patient's titer volume of gas exhaled per breath. And the patient under anesthesia is connected to a ventilator. They don't breathe by themselves. And the titer and volume is something you can adjust using the ventilator. Of course, there is a separate question of the causality, but at least the clinicians will know what drove that kind of a clinical outcome prediction. So importantly, we showed that explainable prediction might improve anesthesiologists' ability to predict clinical conditions in this context. In a user study using a web-based interface, this is to ensure that both prescience and the anesthesiologists were given the exact same amount of information. So what this ROC curve means is that if anesthesiologists can anticipate 15% of the event, with the prescience help, they can anticipate 30% of the event, meaning millions of additional detection of hypoxemia in the US annually, okay? All right, so that was our first application-oriented paper on SHAP. So fast forwarding in time, and then in one of the most recent use cases of this traditional explainable AI technique, SHAP, my student Wei and I focused on the biological age. Our enable age clock or calculator aims to estimate individuals' biological age based on demographic, laboratory examination, and questionnaire-based features totaling 825 features. We trained the model using the UK Biobank data collected from 500,000 people using mortality as a surrogate label. We used XGBoost model, tree ensemble. And here is how it works. So for a 60-year-old person who turned out to be 72 in terms of enable age, the explanations that increased or decreased the biological age come with the extent in the unit of years, which makes it very interpretable. When I said, desirable properties, these theoretical properties of explainable AI methods, this is one of them, additivity. Your feature importance score, you will have the same unit as what you predict. So cancer diagnosis, of course, accelerate biological age, and then also Cysteine C, kidney function measure, which turned out to be important universally, is also, in this case, the factor that increased the biological age. We also identified the mortality specific. So for various mortality causes, we can get this kind of explanations. And then we did a GWAS analysis to identify potential genetic hit for age acceleration. So in this case, the phenotype will be biological age minus chronological age. So in comparison with other biological age clock, different versions of enable age, we show that it more accurately predict 5-year mortality in a separate held out data set we compared with bio-age and pheno-age that are well known in the field. And then we have defined multiple versions of enable age clocks because 820 features, a lot of features. And then for varying combinations of features, we can define different enable age clocks based on the input feature types they utilize. Okay, so more details are in our paper. This is featured on the cover of Lancet Health in Longevity last December. Please check it out. Okay, so among the diseases significantly affecting biological age, we cannot miss Alzheimer's disease. And it stands as a 6th leading cause of death. And then also notably, it is the only top 10 deadly diseases lacking a long-term cure, preventive measures, or treatments to reverse or slow down its progression. This is one sentence I always put at the front of the specific aims of my NIA proposal. So this aspect can be attributed to a limited understanding of early cellular pathways that eventually leads to end-stage pathologies such as A-beta and tau. So in our initial study, a few years ago, my former student, Nick Hasia, first collected the RNA sequencing data from multiple AD study cohorts consisting of both AD and the control samples, and then you will see varying spectrum of the AD severity of this brains, post-mortem brain. And at that point, it was all bulk RNA-seq data set. Then we modeled the 6 AD neuropathological phenotypes based on gene expression levels by using a multi-output neural network model. Here, the goal is not the accurate prediction of neuropathologies. These are all postmortem brain tissues. The goal here is to understand the complex interrelations between genome ID expression levels and neuropathological phenotypes. But again, the key question is the mechanistic explanation, okay? So, and then we show that there are largely two advantages of using explainable AI in this field. Firstly, it enables us to use the neural networks that do not strictly require all phenotypes to be collected in every single sample across all cohorts, which allows us to maximize the number of samples available. And then secondly, the explainable AI is going to provide a post-hoc approach to compute individual genes' contributions to explain each of the neuropathological phenotypes even through this complex black box machine learning model. It doesn't have to be a linear model. And then we showed in our paper that this approach revealed previously unknown mechanisms underlying sex-dependent neuropathology via microglia immune response. Okay? So again, the detailed biological results are described in our paper, the one in the left, Nature Communications. Following that, my former student, Joe, and I delved deeper into the optimizing the utilization of explainable AI techniques for gene expression data input, focusing on two significant enhancements. So first of all, Joe is my former MSTP student, one of the three I have worked with, just got matched to Stanford. And current XAI attempts to explain the prediction based on the input features. However, we know that individual genes are not as interpretable as biological pathways. Some functional units, sets of genes that are involved in the same biological processes. And additionally, unsupervised modeling sometimes gives us advantages. It may enable us to incorporate more training data because we don't have to worry about phenotype labeling. And this is especially advantageous when you deal with neuropathological phenotypes. And in the unsupervised setting then, what does XAI mean? So what does it do? So it can then identify the genes that are important in explaining expression variation within the data set and potentially suggesting their role as drivers. So now Joe can go. So therefore, we designed a new architecture, inherently interpretable neural network architecture that organizes individual genes into pathways within the embedding space. So each latent variable corresponds to a set of genes, predefined set of genes based on pathway databases such as Reactome and KAG. And we also developed the principled method to compute the attributions for these pathways to identify the pathways that are important within the data set, and each pathway's contribution to the input genes to identify potential driver genes. And we applied this approach to the extended RNA sequencing data sets from multiple Alzheimer's disease studies. And then as a result, we identified the mitochondrial complex 1 as an important pathway for AD severity variation. Remember, in this AD cohorts data set, you see varying degrees of AD, and then also consisting of normal brain, control brain tissues, and then Alzheimer's brain tissues. And then that led to the hypothesis of its role as a mediator for tolerance to A-beta toxicity. And then we validated this hypothesis in vivo in collaboration with the UW Pathology using a transgenic C. elegans model of Alzheimer's, and which highlighted mitochondrial complex 1 as a critical mediator for A-beta proteostasis. Is there then a drug that inhibits complex 1? Well, capsaicin is one such example. So after this project, I've been incorporating more peppers into my diet. Thanks. So then we aim to develop an interpretable model for single-cell transcriptomic data sets. So single-cell data sets are routinely collected to study changes in cellular states between control cells and those under treatment conditions, such as pathogenesis, exposure to drugs, infection by pathogens, or genome CRISPR perturbations. And to better understand heterogeneity in treatment condition, it will be important to differentiate latent structure and variations that are uniquely enriched in treated cells from those shared with control cells. So my students, Ethan and Chris, developed a new architecture to explicitly disentangle the data into shared variables, shared latent variables, and then also treatment-specific salient latent variables, okay, that has the activation level of 0 for background cells, okay, and incorporate this idea to the SCVI single-cell variational inference modeling that explicitly models the noise, technical noise from the single-cell data set. Okay, so when we applied this method to the data from drug-treated cancer cells versus untreated cancer cells, we see these cells behave differently in the salient space, depending on their T53 gene mutational status. This is already known, okay, and it's really good to see this. However, it would be, this was possible only because the shared space absorbed this nuisance cell type specific signals, because that's obviously the most, biological dominant biological variables in this data set. And then we don't see this if we use the conventional methods. So, and then we are now continuing this research to understand the single cell level expression signature underlying neurodegeneration. So case control in this case will be healthy control versus AD brains, and then also aging, young and then aged tissues. Okay, so in the second part, we are now explore how explainable AI can catalyze the development of new research avenues. So first, I'm going to show you that explainable AI can transform the way we audit AI systems. So during the pandemic, there was a surge in AI models predicting COVID-19 from chest X-rays. However, they appear to be highly accurate within one data set in a cross-validation setting, but failed when they tested in hospitals external to the training data. And we revealed that they learned shortcuts rather than genuine pathology using our explainable AI technique that can highlight which features were important for that prediction, the saliency map. For instance, it would be right that the model used a clear lung basis as a reason for determining negative COVID-19 status. Okay? But it is not quite right that the model used these laterality markers, the part of this X-ray image, or the medical devices the patient was wearing, pacemaker in this case. And then through many XAI analysis, we basically stopped the field, moved to the wrong direction. There were already 6 published papers, some very prestigious radiology journals, and there are hundreds of related models out there that learned this kind of a shortcut. It turned out that the root cause of this problem was the training data set. So researchers at some point, they used one data source, old data source that has mostly normal, negative, COVID-19 negative, and then combined that with a newer data source that's mostly enriched for COVID-19 positive. So creating almost perfect confounder in that data set. So next year, our paper was featured in really nice Outlook article on the significance of unraveling the black box of AI models. Okay. So this article also included a really nice photo of the 2 co-first authors, the MSTP students. The left, Alex on the left, my third MSTP student, and then Joe on the right, the second one. You will see the first one later. Okay. So our next target is dermatology AI. So we focused on 5 models, including 2 academic models, one published in Nature, and 2 commercial devices, cell phone apps, and 1 competition winner, number 1 in that competition. We saw similar results in terms of the held out data testing. However, when we attempt to apply traditional XAI techniques, we encountered an interesting challenge. You can see that in this case, the saliency map is basically pointing to the entire lesion. It's not very interpretable, not very useful. So clearly, we need to move beyond feature attribution methods. So we developed an auditing framework, AI auditing framework, that incorporates counterfactual images. So the idea is to modify the original image such that it just crosses the classification boundary, in this case, benign to malignant. And in this example, you will see this blue-white veil at the center of this lesion, which normally indicates melanoma. So that way, we will know what this model was thinking. And then we work with the 2 board-certified dermatologists who systematically characterized how such visual features are perceived by each AI model we audited, and then creating humans in the loop. So here's how it works. So for each model audited, we generated many pairs of a malignant versus a benign counterfactuals. And dermatologists then examine these images and assess how the model being audited perceives major visual features. So for example, darker pigmentation, you see one of these 5 systems we audited, it behaves a little differently in terms of darker pigmentation. So when it fails or succeeds, we may know why it's failing. So we can actually dig into the flaws in the reasoning process of these AI models. So this work was just one of many examples where counterfactual AI can potentially revolutionize biomedical AI. And please check out our futuristic perspectives, published in earlier this year. So now our second solution for interpretable medical image AI is to annotate each image with a meaningful concept. Humans don't process image information merely as a pixels. We never view them as a pixels, right? So we think in terms of a visual concept, such as symmetricity, border type, coloration, and so forth. So my student, Chanu, developed an image text foundation model by fine-tuning the clip using 100K image text caption pairs sourced from PubMed articles. And the idea is to bring image and text together into a joint embedding space so that when you have a new image, you can featureize that image based on semantically meaningful concept, okay, with the terms that matter to dermatologists, or not, to identify potential artifacts in the data. And then that can offer an interpretable lens through which we can analyze images. So now next, how does explainable AI contribute to driving cost-aware AI? So here is my former student, Gabe Arion, the first MSTP student, who is now working 24/7 as an emergency medicine resident. He demonstrated during his PhD that explainable AI can enable cost-aware AI in emergency medicine care. So what is it about? So many clinical AI studies emphasize achieving high accuracy, high prediction performance, and celebrating accurate results. However, upon deeper consideration of how these clinical AI systems are used, it becomes evident that gathering features can often be costly in many situations. Say that you have a patient with a traumatic injury in front of you, bleeding, and you need to then predict whether this patient will have acute traumatic coagulopathy, failure to clot. So ATC is time sensitive, requiring massive blood transfusion. So earlier transfusion leads to better outcome. In this case, less organ failure, fewer post-hospital complications, and Obviously, fast diagnosis leads to fast treatment. So accurate prediction of ATC is great, but we really need to predict this fast. And some of the features that need deeper examination aren't the ones you want to rely on too much. So our method, Co-AI, can combine feature importance estimation through XAI methods and feature cost estimated by domain experts to improve both of cost and performance of clinical predictions, okay? So here is the predictive performance plotted against the cost of gathering input features on the x-axis. So in the ATC prediction task, Co-AI can predict as accurate as an existing clinical score, that's a packed score, that's here, but with a significantly lower model budget. In this case, it's a number of minutes. You are seeing the difference between 8 minutes and the 1 minute. And the Co-AI framework is general and then can be applied to various other tasks. So when applied to inpatient mortality prediction in 200 US intensive care units, Co-AI improved the accuracy of risk predictions while reducing the number of features required to make them, and then also outperform many existing clinical risk scores. So this is the area where there are already many clinical scores, especially QSOFA was developed exactly for that purpose. So you want to reduce the number of features as much as possible. Okay, so as the last example of how explainable AI can help us open new research directions, let's talk about cancer combination therapy. As we know, the cutting edge in the treatment of cancers like blood cancer, acute myeloid leukemia, which we focused on, has increasingly becoming combination therapy. And the rationale there is that by choosing the drugs that target complementary biological pathways, we can achieve a greater anti-cancer efficacy. But the issue is that choosing optimal combinations of drugs is a hard problem. There are already hundreds of individual FDA-approved anti-cancer drugs, which means that there can be tens of thousands of possible combinations. If you only consider pairwise combinations, there could be even more if you consider non-FDA-approved experimental drugs in development or consider combinations of more than 2 drugs. So finally, we know that different patients, even patients with the same cancer type, they may respond differently to the exact same drugs. So when we have too many choices, like this case, it would be extremely useful to know why or why not certain drug pairs are not a good option or a good option. And but more than that, our goal in this project was to understand the underlying principles of the biological mechanisms by which any two drugs, have a synergy for a particular patient with AML. So we developed the EXPRESS framework. So explainable prediction of drug synergy. So you could collect a data set with a bunch of samples, including the characteristics of different patients' AML tumor. In this case, we use the gene expression levels, and the characteristics of a bunch of different combinations of drugs, like the structure of those drugs or their biological target genes, and use this data to train a machine learning model. So it takes 2 types of input, expression levels of a patient and for any particular drug combination, drug-based features. And then, the machine learning model is going to generalize so that it can then be used to predict how synergistic different combinations of drugs that you never, tested will be for different patients, okay? But then imagine that it can also generate explanations for the prediction in the form of which genes or which pathways are informative for that particular patient, okay? So that's how Express framework work. So for example, we can consider the feature attributions, SHAP values, for the drug venetoclax, a small molecule that specifically targets the gene BCL2. When we look at the set of genes that were important from this SHAP analysis to the model's prediction of a synergy in combinations that contain venetoclax, we find the actual target BCL2, but also as well as many closely related genes that are known to interact with the BCL2. Okay. So, and then we looked at, what genes are universally important, what kind of genes are universally important across many different drug combinations and patients in our data set. And then we find that two of the most globally important genes are MEIS1 and DLL3, both of which have been known to be linked to AML prognosis, but more importantly, they are related to a cancer level of a differentiation. Or how much that cancer retains a pluripotent stem cell-like state. So the importance of this cancer stemness is further confirmed. We did a further analysis to check what biological pathways are enriched with the genes from the top of our list in terms of the SHAP values. And then we find that the pathway containing genes marking a cell's state of differentiation or stemness within the hematopoietic cell lineage is one of the top pathways. So with many, many analyses to conclude that the stemness could be used as axis to design combination therapies. If you use one drug that's known to work well for less differentiated cancer, you better use the other one known to work for more differentiated cancer. Okay. So now, to summarize, I showed that first, explainable AI involves complex computer science challenges that have required significant advances from the machine learning perspective. We have been in our publishing papers in the ML venues. And these foundational AI advances have demonstrated a significant impact, which is really exciting, across diverse biomedical fields, including clinical medicine, healthcare, cancer biology, precision medicine, and also Alzheimer's disease biology, spanning from basic biology to clinical medicine. So where are we headed now? It has been really exciting to work on diverse problems in both biology and clinical medicine within my lab. And practically, the same underlying principles of what AI can do, what AI can achieve apply to both fields. And which makes it logical to work on both. And then additionally, and more importantly, the future of medicine requires the convergence of both these modalities. So I'd like to see more labs working on both applications. And as we know, AI models are becoming increasingly complex. Nobody's gonna say, convolutional neural network is too complex now. And this means that the pace of advancement in explainable AI must keep up. So our current research endeavors include now broader sense of explainable AI, including concept-based explanations, explainable AI for foundation models, and generative AI models where the notion of interpretability should be refined. I mean, it should even be defined there, right? And then also AI auditing for biomedicine, okay? So again, all the work I presented today was done by my students. I've never generated a single plot or written a single line of the code. And then our research was very generously funded by NIH, NSF, American Cancer Society, and Chan Zuckerberg Initiative. I actually made the sizes of the logos roughly proportional to the size of the support. Don't look too deeply, okay. So, yeah, thank you, but I'm not done yet. So now, before I take questions, I'd like to take a few minutes to conclude with my reflection on my ISCB Innovator Award. An award like this is always given to one among many deserving candidates. And therefore, I believe that the recipient should be given greater responsibility than the privilege. For me, the true meaning of this award lies not in the award itself too much, but in the message I convey from this platform and its impact on the world. As a professor and a scientist, one of the most rewarding aspects is inspiring younger people and influencing their lives. I sincerely hope that my Innovator Award and AI advances I have made will inspire many of these young trainees, and that their aspirations will contribute to solving significant problems in biological sciences, medicine, and society through challenging research. Along the way, I'd like to say to young trainees, always remember the essence of research. The goal is not to publish your papers in prestigious venues like Nature or NeurIPS. The true goal of research and science is to expand the boundaries of human knowledge and capabilities, ultimately creating a better world for humanity. So focus on the future and fundamental values rather than immediate results. Concentrate on ideas, methodologies, and data that most effectively expand the limits of human knowledge and abilities. The results will just follow when you pursue these priorities. If you establish these basic principles, you can genuinely rejoice in the achievement of your colleagues and others, setting your own standards for evaluating research outcomes. You may face various difficulties in research and life. In such moments, focus only on the things you can do and do not waste your time on things you cannot control. At this moment, I think of all the young trainees who might be inspired by my research. The potential of AI in biology is limitless and boundless. And I hope you will never shy away from setting up ambitious goals, not be afraid of challenges, and relentlessly move towards the right goals. No matter what obstacles you face, you will overcome them and achieve your aspirations. Thank you for your attention. And now I welcome any questions you may have.
Thank you so much, Suin. That was a really inspiring talk. And I hope not just the trainees and early career researchers were inspired by that. So we have time for some questions. I think if you want to, Larry's already lined up, so if you also want to ask some questions, if you could head to a mic, that would be helpful. I'll start with a question from online. Thank you for this packed presentation, Dr. Lee. In the Express application combining AML gene expression and drug combination features, I'm curious on what machine learning model or algorithm was used.
Oh, sorry, I try to remember what it was.
It's about the Express application.
Yeah.
What machine learning model was used for?
Yeah, so our priority was first accurate prediction. You want to interpret the model that performs well, right? If the model doesn't perform well, then what's the point of the interpretation? So we first did the accuracy-based, held out data testing, and then identified XGBoost tree ensemble model as the best performing model, and then used the tree-shape to interpret it. And then in terms of the drug-specific features, so this is frequently asked. So we ended up using gene target. Each drug has known, target, and that performed the best.
Thank you. All right, Larry, you're up.
So I want to thank you for the amazing work you've done over a great career, staying true to your values, not being pulled but one way or another by the trends and the, sticking with symbolic stuff and using trees. And I love the auditing work. So first I want to acknowledge the great contributions you've made and how worthy you are of winning this prize. I've really enjoyed watching you. But I want to ask a question about your future directions, because I thought they were really right on, particularly around the interpretability of foundational and generative models. So there's an old-fashioned statistical idea that was used for linear regression called influence analysis. The idea was to try and identify which aspects of the training data influenced an outcome in a particular case. And in some machine learning models, this is trivial. So support vector machines work exactly that way. They tell you which aspects of the training model are at the decision surface for your problem. It would be really useful to understand for foundation models or other really large models with large training sets, which training examples were most important in determining the output. Do you have any ideas about how we could do that?
Yeah, if I had 5 more minutes, I would have elaborated what I mean by foundation model explainability. So our focus is on exactly that topic. So we have been talking about feature attributions in this talk, right? We wanted to estimate the importance of each feature. And then there is also sub-area within explainable AI, whose goal is to identify estimating the importance of samples. So you're thinking about these training data sets that were used for training the foundation model or generative AI model. The question is, which training data sets were important? Say that you are dealing with a single cell foundation model, and that you have some model, and that you want to know which cell types or which cells made the highest contribution. In that case, feature attribution is not going to be very useful. Sample attributions will be useful, right? So there have been methods for doing that uses Shapley values, similar principles. So theoretically well-founded. And then our focus in those newer models, so we are interested in estimating sample level importance. It would be our focus. Thank you for bringing it up.
Thank you so much for this very inspiring talk. I was wondering if you could share maybe your perspective in how would you link explainable AI with fairness in AI for health?
Yeah. So fairness, so it's related to the point I just made, right? So you estimate which samples, which training data sets were important. So fairness issues sometimes are raised because you use the wrong training data. I mean, by wrong, I mean in a fairness sense. So you want to use a training data set that has, say, good uniform distributions of all, but sometimes that's not the case. And how do you even identify that? Say that you have just a model and that you want to know whether that's fair or not. So explainable AI techniques, in particular, this sample level importance estimation could contribute a lot there.
Hi, thank you for the amazing talk. I just have a very quick question. In the model where you were trying to model the different biological pathways, I was wondering if you were just using the pathways as they are from some databases, or you were actually allowing for the model to learn new connections.
Yeah, so that's an excellent question. So this one, yeah. So we used existing pathway databases. We always need to balance whenever we use prior knowledge or existing data sets in a model. It doesn't have to be about explainable AI. When you analyze your data and want to incorporate prior knowledge, you want to do it in a soft manner. You don't want to limit your model to just fix the set of things. So in this case, we used reactome, combination of reactome and CAG pathway. And then the way they organize genes is that for each pathway, they have a list of genes. So it's a gene membership. And then we made the architecture such that each latent variable has a sparse connection with the genes. So each latent variable is connected with all genes that are within the membership. But still, this is a soft constraint in the sense that depending on the learning, after training this neural network, some of the genes may not gain weight. I mean, some of the genes, when you look at the attribution, when it's attributed to back to the gene level, some genes may not even get, the high SHAP value. it could even be, close to 0. So we used this, pathway data set, which may, have, and also cell type specific, disease specific, and so on, right? Yeah, but we made a sparse connection, but still allow the model to learn the connection links with the genes.
Thank you.
Hi, I have a naive question. In your opinion, how this kind of explainable AI will influence the biomarker network areas?
Influence biomarker
Research of networks.
networks.
Yeah.
So I think that in network inference problem, which is in the category of unsupervised learning, right? So you want to learn associations between genes. If it's a gene, GRNs, right? So in that case, explainable AI would be in a slightly different form from what I presented today. It could be that you want to explain the edges. You want to explain the gene I to J, for instance, this edge weight based on certain things, based on some your prior knowledge. We have actually had publications on that. So modeling each, shape value as a function of a certain thing. So basically you are explaining your explanations. So so you just talked about networks. So in that case, you would try to explain network edges.
Yeah. Thanks for your answer.
Sure.
Hi, really, really loved your talk. I had a question about specifically models that use something like images, for example, rather than where the input is a list of features already. I can imagine that it's potentially like a model could find something in an image that goes beyond our current biological understanding, but the pressure to get the explainability would try to constrain that down to what we currently understand about that disease, for example. And I was wondering how you think these competing pressures work in real life and how you consider that in your models.
Between, what you already know versus new things, right? So in this example, so in this Monet example, you can have an image and then you can, I listed in this particular example, small number of concepts there, but you can also look into these medical ontologies and then, get a really long list of terms. And then in our research, we were interested in dermatology terms, but we were interested in something else like purple markers. Sometimes, skin images comes with some marks which dermatologists made. And then these are not medical, the dermatology term, but could be useful to see whether that, training data set is not confounded, right? So you can still, use any terms you want. That's the beauty of this, language model. You can have any terms you like and then featureize any way you want. So you're not really restricting yourself to some small number of terms that make sense. It really, it really depends on what concepts you want to incorporate to your analysis.